we're going to be learning about today and who we're going to be talking to. So I'll turn it over to you, Indu.
Great, thank you. I am excited to see everyone again in our physically distanced but virtually connected universe that we continue to be in. Hopefully everyone's staying safe, and when it's a beautiful time of year in many parts of the country, and I know we have people from all over the world joining us at this point, so it just continues to be exciting. I have been working pretty closely with Mike Okun. As many of you know, we're writing a blog together, and had asked him for some ideas for speakers, and he pretty much gave me a huge number of folks that work with him. It seems like he's picked the brightest minds in the world and brought them together at the University of Florida. And so Matt is one of them, and we'll tell you a little bit about him. So he's a professor of neurology in the neurology department, but he's has a background in genetics. He's studied all over the world. He from the UK originally, spent some time in Canada, in my homeland, did some great work in India there, and then ended up now at the University of Florida. And he is a self-titled gene hunter, which is kind of exciting. I've never really met a self-titled gene hunter, so I'm excited to learn what that is and how that affects our patients. I ended up spending a little time over the weekend watching some of his videos and reading a few of his papers, and he just has a prolific background and really trying to make a difference for families and patients, and specifically most recently in Parkinson's disease patients. And he's met a number of families throughout the world, it sounds like, including some interesting stories, and he'll have to share some of those with us, going to family reunions and trying to map locations of genes so that he can teach us more about the genetics of Parkinson's disease. So, I'm so excited to have you today, Matt, and perhaps you can tell us a little bit about your journey and how you ended up becoming a geneticist who's fascinated by Parkinson's patients.
I'd be happy to, yeah. Can you hear me and see me okay? Because I can't see and hear everybody else. And give me some pointers of when you can see my slides and when you can't and things like that. But just to make some introductory remarks, I feel really privileged to be and to have had the opportunity to work in genetics and human genetics and the human genetics of disease and in Parkinson's disease in particular. I've been working in this field for more than 20 years. And if this was in person, I would probably show you a world map, because it's taken me around the world and many times. And actually that might be quite a nice thing to do right now. In COVID times, when everyone's stuck at home and you can't go places, and I can't go places either, to give you a little flavor of how the global scientific enterprise has come together to try and understand the big question, which is why? Why Parkinson's disease? Why me or you? What causes this disease and how we can put it right? The big question of why and understanding causality, understanding what causes the disease, is the most fundamental that we have to answer. Because if we can understand why and understand fundamentally why it occurs at a molecular level, then I think we've got a chance, a good hope of fixing the problem, of halting the progression of disease in people who already have it, and perhaps preventing it altogether in individuals who are more likely at risk of developing the signs and symptoms of disease. So understanding that question why, which is really critical, really important. So I'm going to take you on a bit of a world tour. Please ask questions throughout. I don't know how the moderators are going to do that. They're going to just say, quiet, Matt, quiet.
No, how it works, I'll just tell you right now, Matt. Okay. So I will man the chat. You don't have to worry about it. I will figure out what questions might be relevant for everyone, because we can't take specific questions, obviously, in this amount of time. So you don't have to worry about a thing. You just have to present your slides and teach us all the goodness that you're doing, and really, sort of the floor is yours. If you'd like, you can start with the slides maybe as a starting place, and then, definitely, leave some time for questions, and we'll just do them at the end of your slides.
Yeah, so we've got about an hour for this, and if I can do some presenting, maybe we have a pause in the middle, or because it'd be nice. I think be great for everyone to be able to participate. So I'd be really keen on that. The genetics of Parkinson's disease is a very big area now. And when I started 20 years ago, there wasn't any. In fact, I remember John Marler, who was the head of the National Institute of Aging intramural program, coming to visit Mayo Clinic where I was working at the time, and I just put in my first big grant on Parkinson's disease genetics, and he said to me, you either know something that everyone else doesn't, or you're remarkably foolish for going into this enterprise. And it really pulled the rug out from underneath me. I thought, what am I doing wrong? And he said to me, Matt, you're a geneticist. You're trained as a geneticist. Why are you working on a sporadic disease that medical textbooks, and I could pull one off the shelf, first chapter on Parkinson's says, there is no genetic etiology in Parkinson's disease. Why bother? And I thought, this is going to be a really difficult proposition, a very difficult challenge to break into this world. But, you know, I just have to look at my family. I look at my parents. I look at my siblings. Now I look at my kids, and I can see why. I can see the family face. I can see the behaviors in my children, which are just like the behaviors in my wife or in me. I can see so much that is seemingly sporadic in terms of these traits, but they're familial. There's so many things about us and the way we behave, predisposition to health and to disease that is genetically encoded, even in sporadic individuals. Genetics is the blueprint of life. It's the backbone. It's a bedrock, if you like, on which all other science is built. However, it's a young discipline. You know, genetics has only been around for probably 100 years, since R.A. Fisher, a little earlier than that with Galton and a few others, and Mendel and his P's, but it's a relatively young science. But nevertheless, it is the bedrock. It is that genetic blueprint for all of the rest of quantitative biology. And it's exceptional in that because it does address and answer that question, which is why. And I'm going to point that out and take on a little bit of world tour. So I'm going to open up my slides.
Perfect.
Can you see these all right? First one's just black and white. Is that showing up?
Quite yet.
Uh-oh.
Andrea?
Why? Do you want me to do it again?
Yeah, maybe. I don't see Andrea on here. Let's see.
He had him, he was sharing him right when he got on.
Okay.
I was.
And
Maybe.
try one more time.
Here we go. I'll try it again. I'll share screen. And this one.
Yep. Yay. Okay.
All
Perfect.
good?
Yep.
So I've got about 31 slides here, and some of them are really hardcore science. And I'm not going to try and bother with that too much. I'm going to try and focus more on telling you some of my tales, really, some of my adventures around the world. And I think that might be more entertaining and perhaps make a more lasting impression as to why and what we've learned.
Perfect.
So to start off with, I have to give you a few disclosures. I used to work for Mayo Foundation, Mayo Clinic. So I started out in a variety of psychiatric hospitals and children's homes. And that's basically where I got into this frame of mind and thinking about neurologic disorders and psychiatric disorders and thinking about, you know, there's got to be a better way of helping these folks overcome their ailment than there presently exists. There's very little therapy for neurologic and psychiatric disorders. And that really had a formative influence on me when I was a kid. But anyway, I trained in medical genetics, in population genetics, so statistical genetics, and then I trained in molecular genetics and then medical genetics, so a variety of things. I ended up in Mayo Clinic, and they patented everything I ever did, by the way. So this is my disclosures, because some of the discoveries I made, including leucine-rich repeat kinase 2, including alpha-synuclein multiplication, things like this, Mayo has the patent, owns the patents here, because as Mayo told me when I left, we own you, we own everything you ever did, and we own everything you ever will do. Anyway, on that note, I left Mayo in 2010, and they're a great institution, very well organized, exceptionally good at clinical care. But I moved to the University of British Columbia for 10 years, and now I'm at the University of Florida, and I'm building a clinical genomic kind of program, a center for human genetics for all of the human condition, if you like, focusing on not just Parkinson's, but many other areas too. But my passion is to figure out Parkinson's disease and to try and do something meaningful about it. You can only really focus on one thing and one thing well. And from a neuroscience lab point of view, that's what I do. So I started off here back in 1996 was kind of when I got started. I looked at Parkinsonism, the classical kind of features, the tremor, the stiffness and slowness, and thought about all the different conditions that manifest Parkinsonism. And there's quite a lot, and I've listed them here. As I'm sure you probably know, Parkinsonism is typically due to the loss of neurons, of cells, in this part of the brain here, in the midbrain, in the substantia nigra. And that innervates the striatum, this kind of structure here of the brain. It sends up long, thin, skinny, poorly myelinated axons up to the striatum, and it produces a neurotransmitter there, which basically allows the striatum, where's my mouse gone? There it is. Allows the striatum to integrate signals from the cortex and from the thalamus together to control movement, the initiation of movement. But there's many other forms of Parkinsonism out there that are known around the world, and they're specific to regional locations. And so, for example, garden variety Parkinson's disease, as we all know well, is at the top of the list here, and it's considered what's known as an alpha-synucleinopathy. I'll get into that in a sec. There's another condition which is almost as prevalent, probably, known as dementia with Lewy bodies, which also manifests with Parkinsonism, but not in all patients, but primarily manifests with cognitive impairment and cognitive decline. There's a condition known as multiple system atrophy. I think we've just identified the first gene for multiple system atrophy. It's a very rare form of Parkinsonism. It doesn't respond well to levodopa, to drugs, and there's a lot of autonomic problems in this form of Parkinsonism. So with things like blood pressure, with sitting and standing, with bowel movements, with bladder, these types of things, autonomic problems that go wrong. But there's some really rare forms. There's this rare form here, neurodegeneration with brain iron accumulation, previously known as Halley-Borden-Spatz, which affects children, causes Parkinsonism with Lewy body-like inclusions in the brain. And you can see here, mean age of onset, 5 years, very young. But there's a variety of other forms of Parkinsonism. One that might be familiar right now, I suppose, in some senses, maybe more in vogue, for the worst possible reasons, is post-encephalitic Parkinsonism. After the 1918 flu pandemic, many people who didn't die of the flu basically went on to get Parkinsonism 5, 10, maybe 15 years later. In fact, half of all people who donated their brain to autopsy in the 1940s had what's known as post-encephalitic Parkinsonism, which was defined as tauopathy based on its brain autopsy findings, rather than Lewy body disease, but clinically manifests very similar to garden variety Parkinson's disease. I don't know, have you ever seen the movie Awakenings? It's a movie with Robert De Niro and Robin Williams. It's a wonderful movie. It's set in an infirmary in New York, and Robin's the doctor there, and he's scratching his head, and he's trying to figure out why these patients who are cataleptic, they're basically sitting there mute in this ward, basically what condition they have. They all have Parkinsonism, they have post-encephalitic Parkinsonism, but he realizes this. In the movie, it's a story really about these patients and levodopa and the development of levodopa. But the patients actually all have PEP, post-encephalitic Parkinsonism, in that movie, and and in the book on which it's based. The frontotemporal lobe dementia with Parkinsonism, this is something that we helped map the gene for back in 1997. This is due to mutations in the first cause, and the primary cause is mutations in a gene called tau, MAPT. PSP, there's a whole variety of things, and then there's some very specific ones like Lytico-Bodig of Guam. I mean, I've had the great fortune of going to Guam and mapping, doing high throughput sequencing of patients with Lytico-Bodig and identifying the cause of disease in many of those patients. And we published this back in 2015 in Annals of Neurology, but I'm not going to talk about that today. There's so many stories I could take, journeys to take you on. X-linked dystonia Parkinsonism. This is common in one major island, Panay, in the Philippines. Very specific to that regional location. There's a variety of others too. But anyway, I'm going to talk to you about genetics and different forms of Parkinsonism. I'm going to focus mostly on the garden variety condition. You know, there's a very famous saying that you might know. When it walks like a duck, quacks like a duck, swims like a duck, it's most likely a duck. And it's from a guy called Jacques Parkinson back in the 17th century who made this mechanical automaton. It was a duck. Basically, if you fed it grain, it would poop. and he took it around all the courts of Europe and told people that basically he's managed to kind of create life, and basically that conjured up that saying, when it walks like a duck, quacks like a duck, it's probably a duck. I focus on, for the most part, late onset forms of familial Parkinson's disease that look just like a sporadic idiopathic condition. They manifest the same way, around the same age of onset. They have the same slow, steady demise, the same response to levodopa, the same pathology at brain autopsy. And I look at those families and I try and map the disease genes that underlie, that cause the question why. Why does that family, different family members in that pedigree manifest the condition? I try and find those genes, and then I'm answering the question, how do they all fit together to manifest the disease? So there's many different types of, before I go there though, I'm going to talk about genetic risk a bit. There's many different types of genetic risk. It's not all the same thing. It's like flavors of ice cream in a sense, but maybe that doesn't do it justice. There are very rare mutations that have very, very high effect. So basically, if you inherit one of these mutations, you'll end up potentially with a monogenic, a single gene cause of Parkinson's disease that is hard to escape from, that passes down the family line from one generation to another, and it manifests in every generation potentially. At the other end of the spectrum, there's very common genetic variability that most people have, or likely to have, with a high degree of frequency, that has very, very modest, if incredibly small actually, effects. So if the odds ratio of 1 here is no effect at all, it's the bottom of the scale here. There's a huge number of genetic variants. Actually, there's probably about 50 million genetic variants like this in every single one of us. That helps constitute our makeup, helps constitute why we're not just susceptible to disease, but also resilient from it. Contributes to how we metabolize drugs, for example, that might help make our symptoms better. And there are some variants that are intermediate, actually a large number of what we call low frequency variants of intermediate effect, which we don't understand. We know what they are, but we don't understand how they correlate with disease yet. And we certainly don't understand that critical question, why they might cause disease when combinations of them come together in an individual, and the individual has symptoms. And examples of that are GBA, for example, glucocerebrosidase. And maybe I'll get to talk about some of that. So there's many types of genetic variation too. I don't know if I can put this down a bit. There we go. There's many types of genetic variation. I'll just show a few on this slide. The slides are available afterwards if you want. But a lot of the variants that people talk about, and the majority of them really in terms of number, are what we call single nucleotide variants. Just a single change, a single point change of one nucleotide out of the 3 billion pairs of nucleotides that we inherit. And You get half the set from your mum, half the set from your dad. And many of these positions in the genome are what we call polymorphic. They will have variability, which is quite common in the population, i.e. that 1% or more of the population may have that particular change, that particular variant. Single nucleotide polymorphisms or single nucleotide variants, SNVs. You also have copy number variants here. This is an example of one here. I should tell you first of all what the slide shows. Along the top of the slide here, it's numbered from 1 to X and Y. Here is a picture of the human genome, and this is basically a sequence of an individual. In this case, it's a male, it's got a Y chromosome. It's got half the number of X chromosome reads, but you can see basically every chromosome. It's just fuzzy here because it's at such high resolution. But each one of these little dots, if I zoomed in, there'd be thousands and millions of little dots here, each one representing a read or a small fragment of sequence of A's, G's, C's, and T's that comprise that particular chromosome 1, 2, 3, or 4, etc. And within there, if you zoom in really, 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 like, hundreds of thousands of times, if you like, resolution, you can see these single nucleotide variants. Where's that mouse going again? Or you can see, in this example here, these short tiles here are the reads, and here there's a decrease. There's a pocket where there's only like 50% of the reads compared to the average bandwidth of reads over that position of the genome, indicating there's a loss of a piece of DNA here. I don't want to use too much technical jargon, but one of the copies of that genome from either the mum or the dad is missing here, and there's represented just a blank. It's a hole. And there's also things like expansions. So, for example, I've actually shown an expansion here of a piece of sequence, which is actually a hexanucleotide repeat, which goes GG, CCCC, and then GG, CCCC, GG, CCCC, many, 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 many times. And if it's expanded hundreds of times, it leads to motor neuron disease. And there's many, many examples of repeats in the human genome like this. There's lots of other genetic variability too that we could talk about today, but I won't. In the interest of time and your sanity. I won't get too much into the weeds. Anyway, come back to my story. I was thinking, my motor car here is, I use an engine of a car or a motor, because that's what we're talking about, it's movement disorders and the ability to move, for the most part. I use this illustration of motor cars because this is kind of what I'm trying to do. In 1997, when I started out, we had an appreciation that the motor didn't run, that there was a movement disorder, but we had really very little idea other than a neuroanatomic one or a pathologic one. When you look at a person's brain at the end of their life, we had a really, really little idea of what's going wrong. Not only that, we didn't even really have much of an idea of the parts, molecularly speaking, within the cells, within the neurons, in the part of the brain that are lost. We didn't even have a good understanding of the parts that are missing or not working from a molecular point of view. So one of the things that genetics is very, very powerful at doing is identifying these component parts. And I think to myself, how are we going to fix it? How are you going to put something right? How are you going to come up with a therapeutic that's going to slow or halt the disease progression and persuade people that if they take it over their lifetime that they're going to stay well, not progress? You really need to understand what the motor looks like. You really need to understand from a molecular point of view how the proteins in that cell interact together to allow that machinery to do its job. And we don't even know those fundamental building blocks. We do now. I've labeled them up. So this is leucine-rich repeat kinase 2. I'm going to talk about that. I'm going to talk a little bit about synuclein 2. The rest of the genes I won't really talk about, but to give you some idea, there's now about 40 single gene disorders that cause Parkinsonism, several of them that look just like late onset typical Parkinson's disease that you might, most people have. And then there's about another minimally 92 regions of the genome that contain another 187 genes that also contribute towards risk. So that's quite a big number of component parts now in that motor. And now we've got to figure out how they work together. So here's where I started. So when I first went to Mayo, I met Manfred Munter. and he has a disorder named after him, and a family in Mayo's archives known as the Spellman-Munter kindred. He inherited this problem, if you like, from his mentor, Spellman, and basically these guys, Manfred was retiring at the time, and he was the chair of neurology at Mayo Scottsdale. And he said to me, Matt, and he showed me this old sepia photograph. He'd been trying for the last 20 to 30 years of his career to figure out what was wrong with these folks. The first lady, and she's here in the picture, she's the lady with her mouth open in the photograph with this rather unusual hand posture because she's holding a baby over the water with one arm. And it's because she's got dystonia in her other hand. And her face is reminiscent of the kind of masked face you see with Parkinson's disease. She had Parkinson's disease in her early 30s. Actually younger than that, what am I saying? She was 24 when she came into Mayo and got a diagnosis of Parkinsonism. And many of her sons went on to get Parkinson's disease as well. And Manfred said to me, you know, for years we've been visiting this family out on the farm in Iowa to try and figure out what it is on the farm that's causing this condition. And they told the family for years, right, that it's something in the well water, there's some pesticide, it's something in that environment which is causing the disease, because the medical textbooks say there's no genetics in Parkinson's disease. But Manfred thought maybe the medical textbook is wrong. And so since the geneticist had just turned up on his doorstep, he thought he'd invite me in and share some of this conundrum with me rather, and to try and help figure this out. So this is a copy of the family, and it took me maybe 6 years of effort to try and pin it back together again, because this branch of the family at that time lived not in Iowa anymore, but in Arizona, in Phoenix. And this part of the family over here, which took an awful lot of effort to pin down, lived in California. This part lived in Seattle, and I went to visit this fellow before he died. And it was an interesting night, pouring down with rain. And I'd called up his mum, and with the movement disorders resident and myself, we went to visit. Because his mum said, yeah, you can come if you like. You know, you guys from Mayo, you've been here many times before, and you know, what are you going to do for us this time? And I said, well, you know, before nobody bothered collecting your blood, nobody bothered looking at DNA, even though DNA was discovered and the structure was worked out in 1953. No one at Mayo for all these years had bothered looking at the genetic etiology of Parkinson's disease, because the medical textbook says it doesn't have one. But I said I wanted to do that because it's clear from looking at your pedigree, you know, this is showing males here as squares and circles of females, and then their offspring, their daughter here and her marriage and her 2 children. Through your pedigree, you can see in the black symbols represent Parkinson's disease that you have an autosomal dominant pattern of inheritance. This is a genetic disorder, and we can map it. We can find the gene, and maybe if we find the gene, it's the first step along the road to putting it right, figuring out what it does. And so I turned up at the doorstep of this house. It's pouring down with rain, and there's motorbikes outside, and these Hell's Angels answered the door. And I was like a little bit thinking to myself with Katrina at the time, we should maybe turn around right now and leave, and probably shouldn't pursue this. You could hear a lot of wailing going on inside the house. It sounded like somebody was very upset, and that turned out to be Mum and the brothers here. Some of the brothers, not this affected gentleman. He was very profoundly affected. He sat on the couch, but his brothers took me out the back, and they wanted to know, you know, it was quite the encounter, what I wanted and what I was going to do for them. And I said, I just want your blood. I just want your blood. And it made them laugh. And you know what? They were very generous. They helped. And we did. It took 6 years, but 6 years later, we managed to map the gene. And it took this is an example of, this is known as interface fish, but basically it's a way of painting, you know, seeing is believing. It's a way of painting chromosomes and within white blood cells, and take a white blood cell population basically and label that with a couple of different color probes here. And these probes are for alpha-synuclein. And it turned out that this family and the people with the disease within the family, and it's segregated down the family line, instead of having 2 copies of the gene of alpha-synuclein, like a normal individual here, wild-type person, they had 4 copies. And on their mutant allele, on the mutated chromosome, they have 3 copies in tandem of alpha-synuclein. The normal wild-type gene, but 3 copies in tandem. And of course, they have the normal contribution from their other parent. We found basically synuclein triplications, as they're called, but it's actually 4 copies, segregate down the family line with disease. And that correlates really well with the age of symptom onset. So you can see it here. Oh, actually, this is the, synuclein triplications here. So by the, let's see, for example, by the age of, 70 years of age, if you have a synuclein triplication mutation of 4 copies, your probability of being affected is probably more than, or as high as 90%. By the age of 50 years of age, your probability of being affected, actually no, sorry, triplication, yeah. By the age of 50 years of age with a triplication, your probability of being affected is probably 80%. By, say 40 years of age, your probability of being affected is 50%. We found many of these families, and we also went on to find synuclein duplications where you have 2 mutant copies. Turns out that alpha-synuclein is, of course, the hallmark of idiopathic Parkinson's disease too, from a pathological point of view, because it basically is this brown stain protein here. Antibodies raised to alpha-synuclein, when thrown against brain sections, beautifully light up Lewy bodies, which are the pathologic hallmark of Parkinson's disease. They also light up all this occult pathology. All of this brown is abnormal. These are neurites within the brain that are stuffed full of aggregated alpha-synuclein. This is a high-power view of a neurite section through a brain, and it's full of alpha-synuclein, and that can't be a good thing. We now know that synuclein ascends, or it seems to appear to ascend through the brain. We can't say that definitively in the human condition because, you know, it's very difficult. We don't have a tracer for synuclein that we can follow this in real time. You can only take series from patients after they've died and say that, okay, it seems to have very mild symptoms or even premotor symptoms. The synuclein's just here in the brain stem. And with motor symptom appearance, it's manifesting, the pathology is manifesting that brown pathology in the midbrain. And as they go on, and for more fulminant in their course, and perhaps with cognitive problems, the synuclein pathology spreads in the throughout the cortex of the brain. We also know that very common genetic variability within alpha-synuclein, here it is in chromosome 4, also contributes to Parkinsonism and dementia in pretty much everybody who has Parkinson's disease. Very common variants. And some of these variants, for example, here in this, we did a really large population genetic study. This is a study of more than 2,000 individuals with either Parkinsonism or dementia or Parkinsonism and dementia. And we found that variability in the one end of the gene manifests, contributes to Parkinsonism and motor dysfunction, and a lot of variability, genetic variability at the other end of the gene contributes to dementia. It's kind of curious. Anyway, long story short, now we know the gene, and we kind of know the mechanism in some sense. We know that overexpression of alpha-synuclein is enough to manifest in the disease, and the more you have, the more that it's expressed, the more copies that you have, and we've looked at the expression levels too, the more likely it is you're going to get the disease younger, and the disease is going to progress faster, and the more rapid that individual's demise to death. That led to the idea that basically if you lower synuclein expression, perhaps you can stop all of that, slow it all down. And now there's at least half a dozen companies that are listed here that are in the game with either antibody-mediated approaches, vaccine-based approaches, or antisense oligonucleotide-based approaches to lower synuclein as a therapeutic for Parkinson's disease. And they're in various phases of clinical trials. But it's very exciting to think, because most patients have this pathology in the brain. If you could clear this pathology, will it prevent the disease? Will it slow down its course? That's the interesting question to ask, and there's a lot of hope that it will in many patients. Certainly, I think it will help with the cognitive aspects if they become manifest and stop that synucleinopathy getting into the cortex. There's many families. This is the paper we did from last year. Each one of these lines represents the region of genome, chromosome 4q21, which is implicated. The ones in red here are probands or index cases from families with 4 copies of alpha-synuclein, and the ones in black are with 3 copies of alpha-synuclein. And you can see the region of chromosome 4q21, this is the box here that's highlighted, is actually different in different families. Each one of these lines represent a family. And this event has happened many times over in history, and many families around the world. And I think it's a wonderful opportunity to basically help develop clinical therapeutics, certainly for these particular patients that are likely to work, because it directly assesses, treats and impacts the molecular cause for their disease. Here's my duck. But I want to go on one further because, like I said at the beginning, there's about 40 monogenic forms of Parkinsonism, and I showed you that engine, right? That's at the beginning. There's a lot of component parts to this. I'm trying to understand how it works for the idiopathic condition, for the unknown genetic form of Parkinson's. How does it work together? And my voyage really around the world has taught me that, and it's a hypothesis still in some ways, that it's a problem in vesicular trafficking, synaptic endosomal trafficking, getting technical, but and that's coming from lots and lots of different data and experiences and lots of different genes that we've mapped over the years in different families that look like, for all intents and purposes, garden variety late onset Parkinson's disease. So I'm going to tell you about a fascinating one, which is LRRK2. I came into this about 2000. It was about 2001 actually that it came out. There was a paper from Japan about a very famous family, a very large family with Parkinson's disease that segregated down the family line. It's known as the Sagamihara family. It's from the Sagamihara prefecture, just outside of Tokyo. And it's a very large kindred. And that was basically published in Annals of Neurology in 2002, mapping a segment of chromosome 12q12 that segregates perfectly down the family line with the disease in the pedigree. And the Japanese group that are working on that, a fellow called Dr. Obata, they hadn't managed to map the exact gene. They couldn't identify it, but they did identify a big window, a big region which it was in. And at the time, in my freezer, and also working, I was working with a clinician, Zbigniew Zolek. We had samples, DNA samples from this family, known as Family D, that's lived in western Nebraska, but actually its origins are originally from Germany. And you can see there's many people in the family affected by the disease. It's the black symbols, and it segregates down the family line. And with the help of this family and another one called Family A, which is actually originally an English pedigree from Lincolnshire, we managed to identify the causal gene, which is known as LRRK2 or leucine-rich repeat kinase 2. Now, not only is this a genetic form of Parkinson's disease in what it was otherwise, apart from the family inheritance, clinically exactly the same as Parkinson's disease, garden variety Parkinson's disease, I keep using that term. But it also rewrote the medical textbooks in many other ways. And one of the most profound ways it did that is that 4 members of this family had come to autopsy, had left their brains for autopsy. And Dennis Dixon, who was the neuropathologist I was working with, had examined all 4 of them. And one of the things he noticed that in 2 of the family members, they had Lewy body pathology, just like the pathology I showed you earlier, stained beautifully with alpha-synuclein, You see these Lewy neurites, here they are. You see these intracellular inclusions, Lewy bodies themselves. 2 of the family members had that, but 1 family member with clinical Parkinson's disease all their life, taking the same drugs, related with the same mutation, it's actually an R1441C mutation in the family, actually had tauopathy, and this is actually stained not with synuclein but with tau, a protein called tau, and this is a neurofibrillary tangle. This is a tufted astrocyte. This is a very typical pathology that you found in progressive supranuclear palsy, or you might find in post-encephalitic Parkinsonism. They have this tauopathy form of, they have this type of pathology. They don't have Lewy body disease. We also found a family member who didn't have either of these proteinaceous inclusion pathologies, but just had a loss of neurons in their nigra. And so how can this be that you have these pleomorphic, we termed it pleomorphic pathologies, in one family in which it's the same, presumably the same cause of disease, segregating down the family line, LRRK2 R1441C. We got very excited about this, and what's shown on this picture is basically a section of a human nigra, the region of the midbrain there. The brainstem, it's a transverse slice kind of across here, and this section of the brain here is normally pigmented. It's normally contains neuromelanin, and it's normally dark in color. But in patients with Parkinson's disease, there's so many neurons that are lost here in cell bodies that it loses its color, becomes pallid. And but it's on the background. I don't know if you recognize this, but this is actually a picture I took of the Rosetta Stone from the British Museum in about 2003. Because it occurred to me, it's a bit like the Rosetta Stone. It links together this one genetic finding, although it's fundamentally important for Parkinson's disease, links together the different hallmark pathologies of many different neurodegenerative diseases. And if we can figure out what LRRK2 is doing in the context of Parkinson's disease, maybe it has application, it will have application to all these other neurodegenerative syndromes as well. Like PEP or progressive supranuclear palsy, or there's many different forms that I showed you on the first slide. It turns out the Sagamihara family, the one from Japan, they had 6 autopsies from that one pedigree, and in those 6 autopsies, all 6 individuals had this. This is saying with tau again, they had tangle disease, and since this time, more than 70 patients now with different LRRK2 mutations, but 2 or 3 very common ones, G2019S and R1441CG mutations, have been examined, and it turns out that half of all patients that come to autopsy with LRRK2 mutations have Lewy body disease, and half have something else. So that rewrites the textbooks as well. One of the most wonderful adventures that I had was actually going to Norway. I met this guy, and his name's Jan Asley. He's a neurologist, and he works up in here, Trondheim, which is, I can just about see it with my glasses on. There we go. It's in central northern Norway. And I met him in a meeting actually I was in Japan, talking to those Japanese investigators, and there was actually a congress in Japan at the time. And I met him at a meeting there, and I was rather disillusioned at the time, because despite the work on alpha-synuclein, that discovery wasn't made until 2003, and this is back in about 2000. It was around 2000, and I had a hard time persuading NIH. I had a hard time persuading Mayo that studying the genetics of Parkinson's disease was worthwhile, and it really wasn't until these discoveries were actually made that they realized it was worthwhile. So I met Jan in 2000, and I was kind of sad. Sitting at the meeting, I was drinking a cup of coffee on my own, and I got chatting to him, and I said to him, none of the buggers at Mayo, all those epidemiologists, want to trust me with any genetic samples of DNA or clinical information, because they really don't believe in what I'm trying to do. And I think I might be a bit of a fool and a bit misguided here. And Jan said to me, Matt, I have some samples from Norway. Would you like them? And I was like, well, that would work out quite well, really. I mean, he was more than willing to do the clinical workup of the families and of the patients within them. He didn't want any funding, and I thought, well, I could probably get funding. Maybe it would de-risk the effort from NIH just to do the genetics, and it could be a great partnership, and it's proven to be the most wonderful partnership. We were fishing at the time for trout, but what we found actually in this region of Norway along the northern coast here is a whole series of families, and I'd already started working them up with Jan, genetically speaking, in the early 2000s. And in 2003, 2004, when we discovered LRRK2, through sequencing these families, we found they discovered they had LRRK2, a mutation in LRRK2 that segregates down the family line in all the pedigrees, known as G2019S. So it's a glycine to serine amino acid change. And at the time, we described it in 13 families, I think 7 from Norway listed here. And you can see some interesting things about the families, right? Not everybody gets the disease in a dominant pattern of inheritance. You expect half of each generation to get the disease and half of each subsequent generation. And so an example like this one, where you've got, mum's got the disease up here, but there are like, 8 siblings in this sibship. Only 2 of them have got the disease. You'd expect 4 of them to have the disease. Maybe that's because some of these family members aren't old enough yet. The disease is age associated, just like late onset typical PD. But it seems to escape some family members altogether. So here, for example, this fellow died. Can't really see it very well, but there's a line through the symbol. Maybe that's why he didn't manifest Parkinson's disease, but his sons both did. Anyway, it's not fully penetrant. That's one of the things we realized. Another was is this. It means a lot to me, but it doesn't mean much to anybody else. But what it is, it's a list of markers of genetic positions along chromosome 12. Right in the middle here is the mutation. It's this G to A change. Here it is, 1427. Everything's got a number, 263. This is the mutation that causes Parkinson's disease. This single nucleotide change from a G to an A at position 6055 in this particular segment of the genome is responsible for these individuals getting PD. It segregates on a larger piece of chromosome, around 200 or so kilobases in size. It's labeled up here in kind of a yellow color. And all of these individuals, although they're all from independent families along the northern central coast of Norway, they all have the same segment of DNA. They all have the same genetic fingerprint. They're all related. And it turns out all these families actually indeed are all related. We've mapped them all back to a 15th century founder. I should keep an eye on the time. Oh, I'm going to run out of time, so I better stop. I better keep going real fast then. They're all related to a 15th. Are you still with me, by the way?
Yeah, we are.
Oh, good.
I wanted to make sure that you got a chance to answer a couple of questions, though, too, because I know you want to make it a little interactive. So if you want to.
I do. So I will do. I'll finish up real quick and then leave time for questions because, it turns out they were related, but they were related also to these other family members, other families. Some of these were from Poland, Ireland, the United States, and it gets even more intriguing. It's a kinase. It activates the kinase. This is the actual mutation itself. It's there's tons of other genetic variability. I didn't take you here, but my journey took me to China. It actually took me to Taiwan, not China, and to the National Institute in Taiwan. And we didn't find LRRK2 G2019S there, but we found a similar variant with a similar haplotype, which is conserved and which is found in everybody with the variant. It's a G2385R. You can date mutations. It's a bit like radiocarbon dating for fossils. You can date them. And this particular one in Taiwan and actually throughout Asia, G2385R is related to one founder about 4,000 years ago. G2019S is related to a founder about 3,000 years ago. We found it spread actually it emanated and started in North Africa, probably in Carthage. It was spread by the Phoenicians throughout the Mediterranean basin and throughout the world. This is G2019S. But I won't take you through it. There's a ton of functional biology. I think I better leave time for questions. Where this goes, just to finish it off, is we can use these insights to make models of gene function and gene dysfunction. This is actually a mouse brain sectioned And these are D1 and D2 neurons, medium spiny neurons in the striate, and labeled up in different colors. And this is on the background of a G2019S mutation, or on the background of knocking out the gene altogether. We published this paper in 2014, but you can actually see what I call accelerated synaptic senescence. The brain of these mice and its function, the candle is burning twice as bright but half as long. That's how I like to think of it. The firing potential of these neurons is much greater in these mice than it should be when they have the G2019S mutation. That's kind of illustrated here, but I won't go into it. We also have the ability to be able to image individuals and look at their neural system 20, 30 years before they even manifest symptoms. The youngest patient in this set, this is by positron emission tomography. The youngest patient is 22 years of age. He's a snowboarder. He's fit as a fiddle, but he's probably going to manifest the disease in 30 years' time. And we're starting to understand how these different components fit together. I won't go there. It's the same. I could talk to you for hours, but I better stop. Is there questions?
Yeah.
If not, carry on talking.
Yeah. Well, that was really. Maybe we could just we can stop sharing for a second, and yeah, there you are. That was really amazing to kind of take us on a journey. I mean, I think what you showed us is that first of all, there was a real, a fight to even get this type of work funded, and you were told you were crazy. And I had watched something where you were talking about this Iowan family where they were had been on a farm and all left and tried to basically just like leave because they thought there was either something infectious or a contaminant on the farm. But then when you really started to sort of look at it, it wasn't truly an environmental thing or an infection, it was these genetics. And so I think really thinking about these models in which we've grown to understand these disease states as purely environmental and thinking kind of a different way. And I commend you for, your strong and work ethic to continue to fight this fight, on behalf of these families, despite it sounds like a lot of barriers. One question that came up in the chat was about specifically Ashkenazi Jewish populations and what types of genes from a Parkinson's perspective they may be at risk for contributing to their disease.
Yeah, so Ashkenazi Jewish populations, they're more closely knit community. For the most part, they have a higher frequency of a whole variety of different genetic conditions, but in Ashkenazi Jews, LRRK2, for example, G2019S, is found in 14% of Ashkenazi Jewish patients with Parkinson's disease, but they also have fairly high frequency of GBA, of glucocerebrosidase mutations, mostly N370S. But what we found, just to take this into a bit of a broader perspective, is there's patches of Parkinson's disease around the planet. We found that there's pockets of it. So, for example, G2019S, although it's common in Ashkenazi Jewish patients, it's commoner still, probably twice as frequent, if not more than that, in Berbers in North Africa. And we find other mutations like R1441G, another LRRK2 mutation. It's only found in the Basques. It's also found in Reno, because I think it's Basque people that have emigrated there. And there are other forms. The Asian mutation I talked about, G2385R, is Asian specific. You don't find G2385R outside of Asia, but you find it all the way from Taiwan, all the way through China, up through Japan and into Korea. It's a patch of Parkinson's disease. And LRRK's got a very specific, the reason for that is because LRRK's involved in immunity. It's involved in maintaining good immune health, and selection for a number of these variants has occurred, evolutionarily speaking, and has driven those variant frequencies from being, the one first mutation that first occurred in one person who originally had it, has driven that frequency of that variant. There's been basically some survival advantage earlier in life. It's a bit like sickle cell trait.
Wow, I didn't know that.
Yeah, really... and so there's patches of this all over the planet. And Parkinson's disease is a manifestation of that in many, many genetic forms.
Right. there's often a question, and I think Rachel actually brings this up, is like, when should your average person who may have, you know, one family member, first degree relative, maybe, you know, that might have had Parkinson's late in life, when should a person be tested for genetic sort of tests? And I know it's probably not a fair question to ask of you because you're, you know, a geneticist and probably you would want everyone tested, but, you know.
No, I don't. I say the opposite. I say in terms of the therapy, there's not much point, really. I would get involved in research, and I definitely contribute to that. And I'd urge you, if you have a family history of Parkinson's disease, or you have living affected family members now, and several of them, get involved in research studies. There are many, many of them going on now around the world. Many, many groups are doing this type of work. But from a clinical point of view, clinical sequencing, it's not really going to change care to know. And yet, when there are, you know, drugs available, then there's a good time to do it. But otherwise, it's a research endeavor.
Okay. And I saw that you had been part of the Michael J. Fox Foundation's Global Genetic PD Study Group. Could you tell us a little bit about that and kind of if you think that patients should get involved and, you know, what constitutes sort of family members with, you know, like, is it 5, 3, 1? I mean, what would be sort of sort of the threshold in which somebody might approach, you know, this type of research?
Well, you know, so I think it'd be a very good thing to do it, even if you have a family history, but you don't have anyone, say, alive, and maybe your parent had Parkinson's disease and now you have Parkinson's disease. It'd still be very valuable to have a genetic sample, a DNA sample from you and your clinical history to add to the aggregate effort to understand what causes this disease. So I'd say get engaged even then. You know, I think in terms of what Fox is doing, you know, they have a very, very big presence in this. They've been amazing in terms of corralling money from pharma, from philanthropy, and basically position themselves between that and the researchers, and help to organize things. And they've been very, very influential there. And now, you know, with NIH, I mean, there's so many other groups as well working on this. With NIH, for example, they have, and with the Brin Foundation, for example, they are driving a global genetic kind of endeavor. I mean, I've been doing it for 20 years, but you know, they are doing much more of the same. They're doing genome-wide associations around the world in different populations. I don't think there's so much focused on families and family-based mapping. There is a GP2 study, which is looking at 5,000 families, multi-incident, trying to understand their disease, trying to map more of these genes. But the question is.
People also ask about, you know, the 23andMe screening, for example, and how useful that might be. What's your sense of patients doing that?
Well, so in 2010, I gave Anwar Cheki a list of variants that she should put on their arrays. And one of them is G2019S, so that's why it's there. But so it does screen for some known genetic forms of Parkinsonism, but certainly not all of them, and certainly nothing novel or new or unique. It only screens for known causes of disease, and it's a very superficial kind of view of it too. I'm a little concerned about 23andMe. I mean, it's a hobby endeavor. It's not in many ways for many people too, and I think they get benefit out of it from that. But I would be a bit careful that, you know, there's no genetic counseling, for example. You can find out whether you're a BRCA1 mutation carrier, whether you're at risk of breast cancer. You know, and this information is provided back without any advice as to what you should do next. You know, maybe that's not what you signed up for when you contributed your sample to find out whether you had G2019S or not. Maybe you don't care because you've got Parkinson's disease and that's your issue, but your other family members might.
Sure.
So I would be cautious with all genetic studies, actually.
Yeah. So it sounds like if you're just, a patient with Parkinson's, what you said, garden variety, sort of like what looks like sporadic, then, just get involved in research in general. If you have known family members that are either alive or passed, getting specifically involved in research that might involve more genetic contributions, maybe through the Michael J. Fox Foundation or somebody like that, that you feel are pretty well connected and doing the right thing. The 23andMe, you think, is kind of just like a hobby, fun,
No, I don't think
issue.
that. It's a private company. It's a private company,
Yeah.
and often private companies do a very good job at pushing an agenda.
Yeah.
But the bottom line is always profit.
Right.
So that's my only caveat really with that.
Okay.
From a research point of view, from getting involved in doing this, you've got to do it from an altruistic point of view.
Yeah.
It's not going to be about what's in it for you or for me or for, it's got to be altruism, because none of these discoveries would be possible unless there's initial finding and then it's validated in another cohort of people. And
Yeah.
many of the discoveries rely on collective effort of tens of thousands of samples being looked at one time versus tens of thousands of control individuals who don't have the disease. It's an aggregate effort. So you've got to look at it as in an altruistic sense, of that you want to do this for the greater good. And it might come back to benefit you in terms of being involved in a clinical trial and things like that, and may find out your LOC2 G2019S and Sanofi Genzyme or whoever, whoever's doing whatever trial may be interested in recruiting you to take part in that trial. And that might be good and all, but it's always research.
Yeah.
It's always at the present time.
Yeah.
It would come on a huge way though.
Yes.
You might be thinking, well, what's the point? It's come on a huge way. In 20 years to go from nothing to 187 genes and 40 monogenic forms and several drugs in phase 2, phase 3 clinical trials, directly precision medicine for Parkinson's disease. It's where oncology was 20 years ago. That's where we are in PD now. And it's all because of folks like you taking part. So I would really encourage it.
Absolutely. I think that's very hopeful and hope inspiring. I think we're just running a couple of minutes late. I was struck by you met with Larry Guilford, who is a fellow Canadian as I am, and did this show for the Global News, and he literally comes and spits in a tube, and you show the exome, sort of the sequencing of his, and explain that, which was really interesting. And we might have you come back and explain that a little bit more. But I was just struck by your words about how long this type of process took just 10 years ago. And you show at some point, he's holding it in his hand, and he's one of the PD Avengers, and he'll be coming on the show, the series in a month, I think, or the next few weeks, to really teach us about his inspiration as a PD Avenger and what they hope to change in terms of treatments and change the field of Parkinson's. But he's literally holding a chip in his hand, and you're like, your entire sequence is on here, and it's pretty remarkable. So I think with teaching us sort of in your eyes, what the changes have been in 10 years and what you see in the next 10 years, sort of just in a few sentences, if that's even possible.
Yeah, so it's really, maybe to take you back 20 years, really. 20 years, it took a decade and what was it? Something in the order of around a little over $3 billion to sequence a single human genome. And today I can do 48 of them in one go, for cost of $400 a piece. It's come on that fast. And it's all because there's been this massive revolution in the ability to do parallel sequencing, and a massive revolution in IT to enable us to assemble all that data, and in cloud computing to be able to warehouse it and manipulate it. And the two things together, this amazing frontier, it's been an exponential, it's been an exponential acceleration in how we can integrate those two things, genetic data and then clinical data. But there's a lot more to discover though. There's so much more to discover. All of those variants of intermediate effect that I pointed out at the beginning, that's most people. what we found is the monogenic ones, the single gene causes, and we know they're causal, absolutely, no doubt about it, but they're relatively rare. And then there's all these common things that everybody has. Doesn't mean to say you're going to get the disease. Everybody's got some synuclein variability that manifests protectional risk, or, you know, of the 187 genes that come out of GWAS, you know, they're very, very common variants. It doesn't drive your destiny, you know. It's that intermediate zone. And that's the bit that's really going, of intermediate variants, genetic variants of modest effect. That's where the action's going to be. And those are going to come through in the next 5 years, like you wouldn't believe. And that, the ability to use machine learning, not that machines learn anything anytime, but the ability to use machine learning to look at patterns. I call it data frameworks. You have a data framework of the patient, their age of onset, their clinical symptoms, their condition, how they manifest, to how the drugs work in them, for example, and how that changes over time. That's one data framework. And you have a data framework of their genome, and you can correlate these two things together now using machine learning for thousands or hundreds of thousands of individuals to see patterns that we couldn't see before. It's a bit like telling the weather. And those patterns basically pinpoint the mechanism, why that person got the disease and what particular type of Parkinson's it is. And of course, leads to the next problem to solve is what are we going to do about it? How are we going to fix it? You know, so we have the icing on the cake. We have the monogenic forms, and we have the base layer, if you like, the crispy crust, the GWAS hits of very frequent but very, very minor effect. It's the bit in the middle that's to come, and it's going to come. Oh yeah. So it needs the engagement of, because it's a machine learning data dredging kind of exercise. It's going to need hundreds and hundreds of thousands of people to get involved. And the more genetic variability there is, the better, because the more easy it's going to be with the big enough sample size to be able to see those patterns and to validate them, to say this particular combination of this variant in this particular gene and this one and this one and this one. It's not just about genes for effect. It's not just about genes for genetic variability that causes disease or manifests risk. It's also genes and proteins that they encode that manifest resilience, that compensate, that mask condition, that mask the symptoms. Yeah. We have many, many families with monogenic disease, with many, many family members who have the gene mutations that we know are causal, who are completely healthy, and they're old, haven't got any disease. But that's because they have a genome that allows them to compensate. We need to find that. We need to find those compensatory genes because those are also targets of therapeutics, you know.
So cool. Wow. Well, this was a really amazing, you know, sort of from start to finish, hearing about your gene hunting. People have thought you should be a Hollywood movie story. They've figured out that Colin Firth should play you, apparently. I don't know if you've been reading the chat at all. And then inspiration of the future, because I think that there's so much progress that we've been making behind the scenes, but it takes somebody like you who has stood by the sort of concept that this is really important and that you should fight to get funding and to continue to collaborate, I think, worldwide with all these people to give you, you know, samples and you know, these discussions. And so I think it's just amazing to hear the story, and hopefully people can get involved and will get involved. And maybe, Matt, you could give us a link or two of, you know, study websites that you think are valid from a genetics perspective that people can kind of go to, because I think there's a huge group of patients that are watching this and will watch it on YouTube that will be open to participate. I think there's a real energy and activism in the air right now. So.
Oh yeah, there's a lot of open source genetic tools and resources and databases, and you know what, it's like I always like it. Maybe it's a bit antithetical. I don't know. I liken it to opening up page 1 of Genesis, right? We've figured it out. We know what there's. We haven't figured out anything yet. We can read we know how to read the text. We can read the A's, G's, C's, and T's now. We couldn't before. We can read them, and now we've got to figure out what it means. You know, we've got to interpret it. It's absolutely incredible. It's mind-blowing. And the thing about it, it's so foundational. Having that genetic insight It's unequivocal. It's foundational for the entire human condition, not just the human condition, for the whole of life. You know, it's amazing. And to be able to read that now, it's incredible. But to be see the patterns and to understand that, okay, this is a word, this is a phrase, this is a paragraph. We're not there yet.
Yeah.
But we're getting very close.
Thank you so much for your time and energy and excitement. You seem to be still passionate about this, even this many years into it, as am I. So I think we'll see where we are in 5 years, hopefully. Thank you for your time and energy. And Andrea, I'm going to pass it back to you for our final goodbye.
Thank you for the invitation.
Yes.
And any time too. There's so many stories, and I really didn't do them justice. I tell you what.
We'll have you back on.
Really fun, really funny, really funny stories. But yeah.
We'll do it. You can show us the chip and all the things that you showed on Larry's episode of the Global News.
Well, I'd actually like to show you the more human side of it. You know, the chips and the technology is one thing, and the IT bit, but actually it's the human stories that are most amazing and so gratifying to me and so humbling. You know, to be given this privilege, right, to be able to understand, to do this work. It's incredible. So I thank you really for getting engaged. And
we'll have you back. Andrea, goodbye wave for all of us.
Thank you. Thank you, Matt. We always show our gratitude with the goodbye waves. I don't know if you can scroll through the box.
I see the wave, yeah.
Everyone,
I don't know if you see mine, but I appreciate it. You know, stay well out there.
Thank you so much.
Stay well out there, you know.
Okay. Take care.
Bye.
Thank you.
Bye for now. Bye.